Hundreds of millions of years ago, the first animals to live on Earth were probably tiny, similar to those we now call protozoa. These animals are all microscopic. Protozoa may be as small as a human red blood cell, or as large as the letter O in your textbook. These are paramecia, swimming among fibers that are about the same thickness as one hair from your own head. Originate? Biologists do not know the answer to this question. However, one theory suggests that protozoa evolved from extremely simple single-celled organisms. Perhaps an ancient plant-like form of life similar to a modern euglena was the first organism to develop animal-like structures such as a light-sensitive eye spot. In a situation where the euglena can choose its environment, the eye spot can detect light, and the flagellum provide a means of moving into the lighted area where photosynthesis can be carried on. In the course of evolution from plant to animal, an ancient single-celled plant may have lost its photosynthetic pigments and yet survived because of its ability to use absorbed food. Such an hypothesis can be tested by experiment. For example, we can demonstrate the loss of chlorophyll in these modern euglena. To do this, we add an antibiotic to culture fluid containing a large euglena population. First, the antibiotic is diluted to a concentration that will not kill the euglena, but only interfere with the euglena's production of chlorophyll. The diluted antibiotic is left in contact with the active green euglena until the pigment, chlorophyll, entirely disappears, as we see by the change in color here. Such animals are incapable of making their own food. Now, since they have lost their chlorophyll, we can demonstrate their need for chemical food by an experiment. We divide this bleached euglena population into two test tubes. To one, we add organic food in the form of a solution of sodium acetate. Food is not added to the other tube. It acts as the control tube. Both tubes are kept in total darkness. After a few days, we find that the population in the tube on the left, containing organic food, has grown, while the population in the tube on the right, without food, has died out. Euglena without chlorophyll, such as we see here, are no longer plant-like, since they cannot photosynthesize. Instead, they must feed upon organic matter to survive. They have become more animal-like. For animals that do not absorb food material from solution, locomotion provides a greater chance of coming in contact with food particles. This Dileptus appears to actively search for food. When it finds food, it very efficiently eats it, as you can see. For convenience in identifying the thousands of kinds or species of the phylum Protozoa, they are classified generally by the kind of structure they use for movement and food gathering. Protozoans which move by means of whip-like flagella are called flagellates. Protozoa, such as the amoeba, move by means of the flowing of their cellular material into pseudopods, or false feet. Some amoebas, such as this actinosphaerium, do not use their thin, rod-like pseudopods for movement, only for the capture of food particles. Some amoebas have hard shells over most of their bodies, but active pseudopods can be seen where they project from a hole in the shell, as in this arcella. Other amoebas have hard internal structures. These skeletons are from marine amoebas called radiolarians. Protozoa like this blue-green stentor move by means of tiny hair-like cilia which beat in a coordinated manner. These protozoa are called ciliates. They control the direction of the beating of their cilia and thereby change direction of locomotion very efficiently. The shape of ciliates varies widely, but all show movement by means of cilia. This blepharisma and this long, lean spirostomum are fast swimmers. In this euplotes, a ciliate, we find the cilia fused together to form the larger leg-like structures called cirri. Other ciliates, such as Vorticella, use their modified cilia to create water currents to bring food their way. Vorticella are attached to solid objects by stalks, which contract when the animal is disturbed. Another group, the Suctoria, are also attached. They capture food with their tentacles, but their stalks cannot contract. They obtain food by chance contact of a ciliate. Then, as their name Suctoria implies, they suck the contents out of the captured ciliate. Some protozoa have no means of locomotion because they have non-motile spore-like bodies. They are called sporozoa. Here we see the sporozoan called plasmodium in the live red blood cells of a chick. If we add a drop of acid to a population of paramecia, as here, the paramecia will move away from the high acidity and congregate in water that is only slightly acid. This shows sensitivity to the... To further explore sensitivity of protozoans to environmental factors, we can demonstrate that paramecia are able to react to differences in temperature. We cool the left side of the glass slide on which the paramecia are swimming. After a period of time, the paramecia have moved to the right, away from cold. A heated iron is applied to the right side of the same glass slide. The paramecia now move to the center of the glass slide, thereby congregating in the region of most favorable temperature. To demonstrate the avoidance reaction of a paramecium, we place them in a tangle of glass fibers. After bumping into an object, the paramecium backs up and moves off in another direction. This is trial and error behavior. The amoeba's reaction to light can be demonstrated quite easily by placing it between bright light and darkness, as in this culture dish, which is half dark and half light. We can see that the amoeba moves out of the strong light into the dark. It is possible to show some aspects of digestion in protozoa by laboratory experimentation. For example, we can demonstrate some of the chemical activity which occurs in a food vacuole during digestion. We will take a small quantity of food for paramecium, such as this yeast. To the yeast, an indicator dye is added. The dyed yeast cells are then added to the environment of a population of paramecia. We will then be able to follow color change in the pink-dyed yeast cells after they have been eaten by paramecia. After a half hour, some of the paramecia are observed under a microscope. In this paramecium from the test population, we can see both pink and red food vacuoles. The change to red color is an indication of digestive activity by the paramecium. The indicator dye added to a tube of plain water shows by the resulting color that the reaction is neutral. That is, the solution is neither acid nor alkaline. This color is comparable to the color of the dye-stained yeast before digestion began. Adding a drop or two of acid will change the color from pink to red. Thus, the red color in the paramecium food vacuole indicates acid conditions in the vacuole. Adding a drop or two of alkali to the indicator causes it to change to blue. Thus, blue vacuoles in the paramecium would indicate an alkaline condition in the vacuoles. To identify these color changes in the paramecia again, let's look at the food vacuoles. We infer that very soon after eating the yeast, the paramecium secretes acid into its vacuoles, thus accounting for the red food vacuoles. Later, the paramecium secretes alkali into its vacuoles, changing the color to blue. In this specimen, the change from acid to alkaline condition is just occurring, as shown by the blue vacuole among several red ones. Other paramecia show blue vacuoles, an indication of further digestion. When a paramecium has grown to a particular size, it will then divide. This is an asexual form of reproduction, since the offspring are derived from only one parent by division of the parent's body. Under favorable conditions, paramecia will divide as many as 4 times a day. If all the paramecia so produced were to survive, it would take only a few days until the mass of paramecia would equal the mass of the Earth. Some one-celled organisms fail to separate after cell division and remain together, forming a colony such as these Volvox colonies. By means of this association, some of the cells specialize for reproduction. Within the larger colonies are daughter colonies, formed by some members of the parent colony that migrated inside the sphere. The importance of the nucleus to continuing life of a protozoan can be demonstrated readily by using amoeba. We support a microneedle on a mound of clay on the microscope. An amoeba in a drop of water on the slide is cut into 2 parts. This half, with the nucleus, will move about and feed. The other, without a nucleus, soon will die and degenerate. Here we see the sexual process of conjugation in the ciliate blepharisma. The 2 animals fuse, and the exchange of nuclear material between the 2 cells occurs. After separation, each animal divides asexually. Most protozoa are free-living, but some have adapted to a parasitic way of life. Some single-celled protozoan parasites cause diseases. Entamoeba histolytica lives in the intestinal tract of man and causes amoebic dysentery. These live gregarines from the intestine of a beetle larva move about in the intestinal content and at some stages of their life cycle invade intestinal cells. Plasmodium causes malaria. The parasites live within the red blood cells of their host. Although a protozoan is composed of only a single cell, it is capable of carrying on all of the basic functions of animal life. It senses, it moves, it ingests food, as this didinium eating a paramecium, the paramecium having eaten bacteria. It assimilates the products of digestion and grows, and it reproduces. Protozoa have been in existence for a greater length of time than any other animal group. They also maintain the largest animal population, and they are adapted to a tremendous variety of habitats.